In this final lecture, we'll talk about how we go about interpreting human neuroscience data and the kinds of inferences that we can or can't make. So historically, there's been two main methods for studying the human brain. The oldest is what we call the lesion method, looking at individuals who've suffered brain lesions due to stroke or other diseases, and looking at how those lesions impact their cognitive functions. This is one example from a particular patient, the patient EP, who suffered from encephalitis that damaged his medial temporal lobes. And this is a study showing that, that EP showed substantial deficits in particular aspects of autobiographical memory. And more recently, neuroimaging has become the most popular technique for understanding and studying the human brain. This is an example of data from a paper by Gilmore and colleagues that looked at activity in the hippocampus, the same region that was lesioned in patient EP, when individuals engage in different aspects of autobiographical memory. The lesion method, originated in the 1800s. This is one of the first examples was by the French neurologist Paul Broca, who encountered a patient that came to be called Tom because the only word that the patient could say pretty much was Tom. And then upon autopsy, Broca found that that person had a lesion in their left inferior frontal cortex. And this actually shows an MRI scan of Broca's brain, which was brought over to the US and scanned by Nina Drunkers and colleague. Here's an example of more recent work. This is from a patient known by the initials SM. This patient suffers from a disorder called Urbach-Beta syndrome that leads to relatively selective degeneration in the amygdala in both hemispheres. And what's known about these patients is that they seem to have a lack of the normal amount of fear. This is a quote from the work by Ralph Adolphs and colleagues, where they talk about exposing SM to live snakes and spiders, taking her on a tour of an open house and showing her emotionally evocative films. On no occasion did SM exhibit fear, and she never endorsed feeling more than a minimal level of fear. Likewise, across large battery of self-report questionnaires and other measures, SM repeatedly demonstrated an absence of overt fear manifestations and an overall impoverished experience of fear. Despite her lack of fear, SM is able to exhibit other basic emotions that experience the respective feeling. So it seems to be relatively selective to fear. Now, if we can look at the logic that's used in studies of lesion patients, in general, it's often referred to as a dissociation logic, or sometimes a fractionation. The idea is that we want to use lesion data to demonstrate that two different cognitive tasks are supported by different brain systems. Now, the simplest thing we might imagine is what's often called a single dissociation, in which you would have, one group, let's call group X here, that's impaired on one task but not another, but then, have a group Y that is perfectly fine at both of those tasks. Now, we can't infer from this that the two different tasks, A and B, reflect different underlying functions. For example, it could be that the task B is just way too easy. And if we had a more difficult task, we actually might see that, that group X would also be impaired at that. So what's more often used is something that's called a double dissociation, where we have two different groups of patients, one of which is impaired on one task but not the other. So group X here would be impaired on task A but not B. And then another group, in this case group Y, that's impaired on task B but not A. And that lets us be more confident that task A and task B are relying on different functions that happen to be impaired due to the lesions in those two groups. Now, this is still challenging to interpret, but this is the general logic that's often used in these sorts of studies. Here's one example, a paper by John Gabrielli and colleagues published in 1995. They wanted to look at the differences between declarative memory, that is memory for previous events, and repetition priming, which is an improvement of performance with practice. And what they saw was two different groups. One, people with amnesia who had impaired recognition memory, but normal repetition priming on a particular task. And a patient known as MS who had a lesion of the visual cortex who showed the opposite pattern, normal recognition memory, but impaired visual priming. And this establishes reliance of different memories on different memory systems for these two different tasks. Now, what we know about lesions is that they can help us establish the causal role of a brain area. That is, if a region is necessary for performance of a particular function, if lesioning it disrupts that function, then that region is causally necessary for the task. But there are challenges in interpreting lesion effects. And in particular, we have to rely on nature to create the lesions that we're going to study. And often they're very large lesions, which can lead to confusion about, what's going on. So for example, it's long thought that damage to the inferior frontal cortex in the left hemisphere was the lesion that was responsible for the speech production deficits that are seen in what's called Broca's aphasia. But some work by Nina Dronkers and colleagues showed that if you look across patients, the critical lesion, that is the place where you have to have a lesion in order to experience that deficit, is actually in the insular cortex, not in the left inferior frontal cortex. Both of these fall within the same territory of the vasculature, that is the middle cerebral artery, and thus when a person has a stroke, they're likely to be damaged together. And so this led to some confusion about what the actual necessary region is, and that needs to be kept in mind. Now, when we talk about fMRI, something that is often said is that fMRI can only provide correlational evidence. This is a quote from this brain metrics piece on Nature Education. And I want to unpack a little bit more why that might or might not be exactly right. So let's think about the causal structure of a task fMRI experiment. Let's say I give a person a Stroop task. I show them the word blue written in yellow ink, and I say, tell me the color of the ink. This task, when I give the person a task, that causes a particular pattern of neural activity, right? So there's causation there by my task. And this causes an increase of blood flow, right? Again, there's causation here that ultimately gives rise to that activation pattern. So it's not fair to say that there's no causality going on. What's more important is whether we can infer the causal impact of a particular region. So first, let's ask, what is it that activation does or doesn't mean? Now, people often infer, because it's called activation, that it means an increase in the level of excitatory neural firing. And that is sometimes the case. But what we know actually is that fMRI signals reflect overall synaptic activity, which includes both excitatory and inhibitory neurons. And so the degree to which fMRI signals reflect either excitation or inhibition is going to depend on the exact nature of the microcircuits that are being imaged. So we know, for example, that there are demonstrations of known inhibitory inputs driving increases, being associated with increases in blood flow. And there are also examples of changes in blood flow due solely to synaptic activity with absolutely no spiking in particular animal models. And so the relationship between activation and sort of excitation versus inhibition is complicated. We also can't infer when we see activation in a region that region is necessary for task performance. And now there's now a number of demonstrations of activation in regions that we know not to be necessary for performance. This is just one example. On the right, I'm showing you data from a meta-analysis by Barcher and colleagues that looked at activation patterns associated with the processing of subjective value, for example, different amounts of reward. And the ventromedial prefrontal cortex is very strongly associated across many studies with the representation of subjective value. However, studies by Leslie Fellows and colleagues of patients with lesions to this area show that their ability to perform the tasks that give rise to activation in this area is at worst minimally disrupted. They don't show the kinds of deficits that one would expect if this area was really the seat of value-based decision-making. So we can't make this kind of backward causal claim about the causal role of a brain activity in a task, given that it's been activated. We need lesion data to help us make that sort of claim. So I want to talk about a couple of other challenges in interpreting functional MRI data. And one is what we've come to call the reaction time paradox. It turns out that when looking at functional MRI data, it's almost impossible to distinguish differences in activity related to the duration of neural activity, that is, how many milliseconds were the neurons firing, compared to differences in the intensity of activity, that is, how strongly were they firing during that period, how many neurons were firing. And this raises a paradox, which is, in the kinds of tasks that we use in task functional MRI, there's almost always a longer reaction time for our kind of experimental task of interest compared to our baseline condition. And in fact, in psychology, those differences in reaction time are actually the effect of interest. However, in fMRI, they're actually a potential confound. This is a demonstration from Tagliarconi and colleagues that showed regions that were correlated with reaction time in a relatively non-specific way across many different psychological tasks. And you see that these are actually the areas that are very often active in cognitive tasks, areas like the anterior cingulate, the anterior insula, and the lateral prefrontal cortex. And so what this suggests is that when we see activation in one of these areas, if we haven't properly modeled the effects of reaction time and sort of removed them from the fMRI signal, then we can't be sure whether this reflects increased activity of the neurons in that area, or simply reflects the fact that the neurons are doing exactly the same thing. They're just doing it for a little bit longer because the reaction time is longer. Now, another type of explanation, or what I would really call a non-explanation in neuroimaging, is the idea of efficiency. And this came first from work by Hare and colleagues in the context of intelligence. This is a quote from one of their early papers saying, a series of investigations indicate an inverse relationship between glucose metabolism in the brain and psychometric measures of intelligence. And those studies have been interpreted as evidence for a brain efficiency model of intelligence. Intelligence is not a function of how hard the brain works, but rather how efficiently it works. And I want to try to convince you now that that actually isn't explaining anything. It's simply renaming the phenomenon. And I want to do that with a thought experiment. Let's imagine that a Porsche and a Prius both take the drive from San Francisco to Los Angeles. They take exactly the same route, but the Prius uses much less gasoline. Now, we can give an explanation for why that is. It has to do with the nature of the engine, the hybrid engine in the Prius. It has regenerative braking, so it can feed some power from the brakes back into the to the power system. And so we can come up with a physical explanation of that difference. Now, somebody might also say, well, why does the Prius use less gasoline? Well, it's because it's more efficient, right? And we would never accept that as an explanation for why it is that the Prius is actually using less gasoline, right? It's simply renaming the phenomenon of using less gasoline. So we have to be very careful about things that seem like explanations, but aren't really explanations at all. They're just renaming the phenomenon. So if we ask, what do differences in activation mean? It turns out that there's a lot of different outcomes that could give rise to any particular difference in activation. So let's say that we see, a difference across two different task conditions. It could be that a different set of cognitive processes is being performed across those two. It could be that the same task is being performed, the same cognitive process is being performed, but a different kind of neural computation is being performed to complete the same cognitive process. It could be that the same neural computation is being performed for the same amount of time, but with a different intensity. Or it could be that the same computation is being performed at the same intensity for a different amount of time. Finally, it could well be that the same neural computation is performed with identical time and intensity, but for some reason there's a different metabolic expenditure between the groups or the conditions. And what's essential to understand is that it's generally impossible to determine which of these is true based solely on the fMRI data. We need additional data regarding either the task or the underlying biology of the data in order to really understand what differences in activation mean. So in summary, if we want to make causal claims about brain function, we require a combination of methods in order to be able to do that. Brain imaging alone cannot establish the causal role of an activated region. And the interpretation of activation differences can be very challenging.